All right, these are going to be notes on transcription and translation, given March 1st of this year. The first thing that we need to talk about is that DNA is used to make amino acids. But DNA can't be directly used to make amino acids, and so we need a stepping stone. Amino acids are the monomers of proteins. Monomers are building blocks. So it takes a lot of amino acids all bonded together in order to make a protein. DNA is used to make amino acids, but it must be changed to RNA first. Because DNA is too precious to leave the nucleus, we have to have RNA, the cheap DNA counterpart, in order to accomplish that goal. If at any point in time the notes are too fast or too slow, you can speed them up or slow them down or pause. So RNA is ribonucleic acid, very similar to DNA, just has a different sugar that it is bonded to, and one of the nitrogenous bases is different. So this process of DNA being converted to RNA is called transcription. Transcription is the process in which the information in DNA is copied to RNA. This takes place in the nucleus of the cell. It must happen in the nucleus because DNA never leaves the nucleus. When we talk about mitosis, sometimes the nucleus leaves DNA, but DNA never leaves the nucleus. And all RNA are made this way. They're made by copies of DNA. In humans, anyway. There is no T in RNA. There is a U in uracil, for uracil. So those are the nitrogenous bases that are switched out. So DNA, the nitrogenous bases are adenine, guanine, cytosine, and thymine. And in RNA, we have adenine, guanine, cytosine, and uracil. And we'll look at that a little closer in detail when we look at translation and why that matters. So T and U switched. Here's a look at the DNA bases, A, T, G, and C, and the RNA bases A, U, G, and C. So adenine, thymine, guanine, cytosine, adenine, guanine, cytosine, and uracil. And uracil and thymine are really chemically similar. The only difference is there's an extra methyl group here, and it is missing in uracil. So thymine has an extra carbon, a couple extra hydrogens. Uracil is lacking that methyl group, that CH3 group. So translation, where we convert RNA to proteins, looks like this. Proteins are just strings of amino acids. The genetic code is broken up into sequences 3 nucleotides long called codons. Each 3 nucleotide codon codes for a different amino acid. So AGT, GGG, and you'd have a long string of DNA and then RNA that looks like this. This is a strand of DNA because it has thymine in it. That would get transcribed to RNA and then translated to amino acids. Excuse me. Certain codons signal to stop and start as well. So certain 3 nucleotide sequences code for an amino acid, and other 3 nucleotide sequences code for either start making a protein or stop making a protein. Now let's look at this. If we convert DNA to mRNA, that is transcription. We can convert mRNA to amino acids, that is translation. So here are example tiny segments of DNA code. The way we would convert to mRNA is by remembering that all tigers can growl. What that means is that A always bonds to T, and C always bonds to G. But remember that in RNA, there is no T, there is a U. So you could think of this as all unicorns can growl if you're converting from DNA to RNA. But you need to leave this T because if there is a T in DNA, like here and here, those still need to be converted to A's. Only if there is an A in DNA, like here and here and here and here, will those get converted to U's in RNA. So here we have A goes to U, G goes to C, and T goes to A. So A turns into U, G turns into C, and T turns into A. Next up we have TAG. TAG will get converted because T bonds with A. A ends up being a U in RNA, and G bonds with C. So remember to keep those nucleotide chunks together in order to make this work. The last thing we'll do is convert to amino acids. So I need to look for UCA, and where I'm going to look for that is right here. So the way you use this tool is the innermost section is the first nucleotide in your 3-nucleotide sequence. The middle section is the second nucleotide, and the outer section is the third nucleotide. So we had, let me check again, UCA. So I need to look at U, and once I find that I'm working with that U, then I know that I only care about this quadrant. Next up was C. So there's a C, so now I only care about this section right here. And then whether it's UCU, UCC, UCA, or UCG, doesn't matter. We're always going to end up at serine. So I can safely say that that first amino acid is going to be serine. All right, those are the notes on transcription and translation. These will be more practice. I'm going to solve them now, but you can solve them on your own, and then reference this video. So pause for a moment if you'd like to try and solve those on your own. There you are, completed solve. All right, send me an email if you have any questions. Take care.